Hello everybody. Today I will talk about the discovery and implications of DNA protection and rejuvenation effects of DNA gaps. These two terms are new terms for everyone, but actually we have been studying and discovered the DNA gaps since 2008, and we have been knowing the role of DNA gap as DNA protection and rejuvenation actually recently. That's why it is new to everyone. The DNA gap, the full name of DNA gap is Youth Associated Genomic Stabilization DNA gap, because the DNA gap has function in stabilizing DNA or genome and can be found in youth DNA more than aging DNA. So because of, you can see at this term, we think that the function, the mechanism that our DNA age is the reduction of DNA gap. As we are proving in this picture that if we add molecules that produce DNA gap, we can rejuvenate DNA and rejuvenate the cell and whole body by stabilize the genome. And then I will discuss how the finding of DNA gap explain the aging process. You can see from this slide that in rat liver, in aging rat liver, you can have a lot more number of senescent cells which stand in blue. But after we add DNA gap, by adding regime, the number of senescent cells reduce back to the youth. In addition to senescent cell in liver, everything else in our experiment also rejuvenate as well. One of the theory that may explain aging process is that in aged cell, DNA damage increase. As you can see in vitro experiment or in other models, DNA damage is the key molecule to cause senescence. But in aged cell, nobody knows how DNA damage increase. But the other thing that we see during the aging process is that some of the epigenetic marks decrease. So in theory, epigenetic marks may protect DNA from being damaged. So reduction of epigenetic marks may lose DNA protection effects. Then DNA damage spontaneously increase in the cells. So if this theory is true, if this hypothesis is true, then adding epigenetic marks into the cell can rejuvenate the whole cell and the whole body. So the study, our study showed that in our DNA, which is double-strand DNA, when the double-strand DNA function by mRNA synthesis or DNA replication, one process has to occur is that denaturation. The denaturation, because of the DNA structure, is double helix. So the degeneration will cause spinning of the DNA. Without, if the end of DNA is closed, the spinning of DNA will cause DNA tension and DNA damage along the line of the process. What we found is that our DNA, particularly young DNA, has DNA gap, so that when the DNA function and degeneration occur, the spinning of DNA tension will relieve from at the DNA gap. But in aging DNA, the DNA gap reduces, so that when the DNA denature, the spinning of DNA causes a lot of DNA tension and DNA damage at aging DNA at the end. So in this talk, I will show you that by forming DNA gap by epigenetic editing, particularly adding molecular scissor, we can rejuvenate DNA. The detailed process of molecular scissor in rejuvenating rat will be presented by Dr. Yasom in this conference as well. At first, we also asked the hypothesis of our first paper did not think that we are looking for DNA gap. We thought that we are looking for pathologic endogenous DNA double-strand break because a lot of study demonstrated that hypomethylated genome will have accumulate mutation easily, and a lot of mutation are DNA deletions. So the mediator or intermediate form of DNA deletion would be endogenous DNA break. So our hypothesis at that time would be that if we measure DNA methylation of endogenous DNA break, we will see the methylation of endogenous DNA break would be hypomethylated. However, when we measure methylation of DNA break, we see opposite result. In this graph, our hypothesis expect every DNA result supposed to be localized here. Surprisingly, it's localized here. In this graph, the x-axis represents methylation of the genome, and y-axis represents methylation of DNA break. So if DNA break were hypomethylated, the result is supposed to be here. But every result is localized here, meaning that every DNA break, every endogenous DNA break that we detect were hypermethylated. The other surprising result is that we detect DNA break in every cell, including non-dividing cells and cell in T-cell phase. In theory, that if it is pathologic break, it's supposed to localize it only if supposed to be detectable only in S phase. So what we see here is not pathologic break. So if it's not pathologic break, the other thing opposite to pathologic break would be something such as epigenetics. So if it is epigenetics, then it's supposed to be produced by our cells. So we study DNA gap levels in mutant yeast. If we can see DNA gap level in mutant yeast that reduce, meaning that mutant may be the proteins or genes that produce that DNA gap. You can see from the picture of experiment, we also be able to measure DNA gap in yeast, meaning that this is a DNA structure, DNA modification that are conserved since yeast to human. The second thing is that some of the mutant reduce DNA gap. The first group is HMGP group, and the one that we will talk about is NHP6A, which is represent HMGP1 in human, and the other one is SIR2, which represents SIR1 in human. These two genes are play many important roles in human physiology and pathogenesis. So DNA gap are produced by our genes. So if it is produced by our genes, it may have biological roles. To study biological roles of DNA gap, we first using yeast model and prove that the biological role of DNA gap is important in aging process. So we measure the level of DNA gap in chronic aging yeast, and you can see that the more yeast age, the reduction of DNA gap occurs. Along the reduction of DNA gap, you can see that the association between DNA gap level and viability of the cells. So this slide shows association study that the role of DNA gap may be important in preventing aging process. So we study more, try to determine the role of DNA gap, because the previous study demonstrated the aging and the role of DNA gap in aging. We have to have a yeast model that has different level of DNA gap but have the same age. So we select to use HO induction. The first experiment represents the accuracy of our experiment, that DNA break that or DNA gap that we discover was not because of DNA preparation, because we show the same pattern of intranuclear preparation as well as the same as high morphology DNA. So when we reduce DNA gap by HO induction, what we saw is that we also reduce viability of the cell as well, showing with these two graphs. So this is the same thing as we show in aging cell. So the DNA gap has something to do with viability of the cells. And the hypothesis is that, as we show in the previous picture, DNA gap may prevent DNA sharing because it reduces DNA tension. So we prove that DNA sharing occurs by adding caffeine into the cell to show that the caffeine prevents DNA repair, and we measure the increasing of pathologic break. The slide shows that in HO induction cells, pathologic break increase a lot more than in HO induction cell than control cells. And then we also confirm this phenomena in NHP6A deletion, that pathologic break occur a lot more in NHP6A deleted cells. And we sequence DNA gap and DNA break, the whole genome, and you can see that the red bar represents pathologic break. The blue bar represents the red bar, and the green bar represent pathologic break. The blue bar represents DNA gap, and you can see that in cells with reduction of DNA gap, after adding caffeine, you can have a lot more pathologic break in the cells. So we prove the function of DNA gap, that DNA gap has a function to prevent pathologic break. So in this study, we prove that DNA gap actually epigenetic marks, not DNA damage. Because if you review epigenetic mark and DNA damage and compare it, for example, DNA methylation compared with DNA damage type of 8-hydroxy-DG, you can see that DNA methylation occurs by cellular enzymes, and 8-hydroxy-DG occurs by oxidative stress. And DNA methylation has a lot of function in physiologic functions, for example, gene control and genomic stability of the genome. But 8-hydroxy-DG has pathologic effects, range from mutation, apoptosis, and senescence. So for DNA gap are produced by HNGB group and be maintained by SIR2. So it's produced by cellular enzymes, and it prevents DNA sharing, so it has physiologic function. So youth DNA gap is a new epigenetic mark. In our current study, we test whether the role of HMGB1 and SIR1, that HMGB1 produced DNA gap, and SIR1 function is to deacetylate histone to form heterochromatin around DNA gap to prevent DNA gap from detection as a pathologic break. And the function of DNA gap is to prevent DNA damage along the ring of the genome. In yeast, we prove that it prevents DNA pathologic break, but in human model and in rat model, we prove that it prevents every kind of DNA damage. These are two examples. The first example of proving this complex, that when we add TSA or reduction of 30 in 1, the DNA gap change to pathologic break. And we add HMGB1, part of HMGB1 to produce DNA gap. But instead of production of DNA gap, the DNA gap under the cell with TSA or without 13-in-1 was recognized by gamma H2AX, which means that this produced DNA gap pathologic break. As you can see from the example of the cell, the signal is quite clear. The second experiment, we proved that if we add DNA gap to the cells, we can stabilize or increase DNA durability of the genome. The picture shows the cells after X-ray, and you can see the H2X foci after X-ray compared with cell with increased DNA gap. I think this is the only technology in the world that can increase DNA durability. In conclusion, DNA gap functions as the gap similar to the gap of the railway track. That you know that in railway track you have to have gap so that when the temperature of the environment increasing, the DNA gap will prevent the torsion of the railway track. The same thing will happen to DNA gap. Here is that DNA gap prevent torsion of DNA, particularly in the youth, but in old age, the DNA gap reduced so that the tension of DNA increased and the aging occurs. This is the whole theory of aging that we think we can conclude from our study. People know that if you have DNA damage, then you send a signal to arrest growth and enter senescence. But people do not know how aging DNA increases DNA damage. We prove that reduction of DNA gap can increase DNA damage. So we think that the reduction of DNA gap is the nidus process of aging. And I summarized the result of the rejuvenating rat here and predict the implication of the study in the future, to urge everyone, to emphasize everyone that this is very important and you may want to join the study of DNA gap. After rejuvenating rat, we can completely rejuvenate, reduce senescent cells, increase liver function, recover memory, and reduce all senescent associated protein, reduce visceral fat. More importantly, we can reduce liver fibrosis back to normal as well. So the potential of reduction of senescent cell and also fibrosis lead to immense a lot of application in clinic and even in agricultural products. In addition to rejuvenating rat, we also used red gem to create the burn wound and also DM wound, and we also improve quality of pork meat. The animal experiment show the promising future clinical application of red gems, which is rejuvenating DNA by genomic stability molecules. The function is to produce DNA gap, as we call it, molecular C-cell. So we think this happens in aging process. If you review literature, you know that the early event of aging is HMGB1 released, and HMGB1 has been studied as a cause of inflammation, or actually as a signal of white blood cell to get rid of senescent cells. But the other thing what that happened is that what happened would be reduction of HMGB1 in nucleus. And when you reduce HMGB1 in nucleus, you will have urinary gap reduction. When you have urinary gap reduction, you will have DNA damage. And then DNA damage will signaling DDR, which is DNA damage response, and persistence of DNA damage response will signal cell to enter senescence. The actual process, I think, is something like reduction until it reaches threshold, so cell can enter senescence. So aging process is quite difficult to reverse by naturally because of lack of HMGB1 in the cell. But molecular sisters imitate the HMGB1 process by producing DNA gap so that we can stop DNA damage accumulation and rejuvenate the cells. Thank you very much.